Good afternoon, everyone. Am I audible to everyone? Okay, this is Banaja Prakashini Samantarai from Central University of South Bihar, and I'll be presenting my work on multigenerational effect on survivability of Xerodaphnia cornuta to Mancozeb. I would like to start my presentation by thanking the organizers for giving us coffee after the lunch break, which is why I can expect that people will be listening to me, even though this is like the last session. So, I would also like to thank Dr. Prasad, who gave his talk on transgenerational plasticity, because of which I can expect a little bit of my foundation work that has been covered. So I can quickly move to my topic, which is, okay, it's about agrochemicals. So first of all, we are aware that agrochemicals are the pesticides, fertilizers, fungicides, weedicides which are used for agriculture. And the agrochemical that I have used is Mancozeb. It is an ethylene-based dithiocarbamate, zinc and manganese salt, and it has been recorded in various aquatic environments. Now, I would like to say that Mancozeb is a widely used fungicide in India, and its usage has been recorded to be around 1,000 metric ton or maybe more. It is yet to be banned in India. So moving on to, can anyone tell me how many of us are aware about zooplankton? Can I see any raise of hands? Okay, that's more than what I expected, but still I'm glad that I have this slide. So, zooplankton, they are aquatic microorganisms. They are found in both freshwater and marine environments, and they are very important part of the aquatic food web. And they also serve as food for higher trophic organisms like the larva of fish, and for higher zooplankton themselves. They mostly feed on algae, but they are also carnivorous in nature, so we can find both herbivorous and carnivorous zooplankton. Moving on to my test organism, it is Xerodaphnia cornuta. How many of us are aware of Xerodaphnia cornuta? Okay, only one. How many are aware about Daphnia? Okay, so the normal Daphnia magna, which you will find in many research, it's not found in India. We have Daphnia, but we have different species of Daphnia, but the Daphnia magna has not been recorded in wild in India yet. If anyone finds it, please let me know. So this is the tropical counterpart of the Daphnia. It is Xerodaphnia cornuta. It is a crustacean, it's a cladocerae, and it is a parthenogenetic female, which is interesting because a parthenogenetic female is basically all Xerodaphnia that you find in a favorable environment. They are females. They produce eggs, not exactly eggs. They give birth to identical females, and they only have males in unfavorable conditions, which leads to resting eggs. So to start an entire population of Xerodaphnia, you only need one, and that's usually called a clonal generation, and these are the clones that we are going to use for our experiment. So for the methods, first we sampled zooplankton from a local pond, and then we isolated our Xerodaphnia, and then we cultured it in lab conditions, and we added Chlorella vulgaris, which is a single-celled algae, to serve as food. Then we had an acute toxicity test where we subject the organism to a series of concentrations to find out that which is the lethal concentration for Mancozeb. It is a standard ecotoxicology procedure, and we used OECD guidelines, and we had the observation for 96 hours. This followed a chronic toxicity test where we subjected the Xerodaphnia to 6 different concentrations of Mancozeb with along with control, and we take the observations till the last organism is alive. All observations are taken in every 24 hours, and the test media is also refreshed in 24 hours, and we did the life table parameters assessment. Coming to the multi-generational exposure, we have exposed the Pseudodaphnia cornuta to 6 generations, and most of it, the organisms that were produced, or the neonates that were produced from the F1 generation, served as the starter organisms for the F2 generation. We have taken organisms for the fourth and the fifth brood only, because according to literature it is said that the fourth and the fifth brood organisms are the ones which are fittest. All the experiments were done in 5 replicates, and there were a cohort of 10 organisms each. Coming to the chronic toxicity test, this is the graphs for the age at first reproduction, generation time, life expectancy at birth, and the second graph is for gross reproductive rate and net reproductive rate. You can see that with the increase in concentration, there is a decrease in, well, at least there is a clear decrease in reproduction. For the life expectancy at birth also, you see a clear decrease, and the generation time, though, it is also decreasing. The age at first reproduction, that is what is increasing. Moving on to the multi-generational exposure. Actually, one thing I would like to point out, from all the test concentrations that we have used, we have seen the minimal effect in 1.54 microgram per liter, which is our test concentration, because this is also equivalent to the concentration of mancozeb or its metabolites that have been observed in the nature. In the multi-generational exposure, we see that there is a clear, it is clear that in the control there was not much variation, which is why the ANOVA for the control, it was also not significant. Moving on to the treatment, we can see that there is a rise in the life expectancy at birth, suggesting that there is some increase in the survivability of the organism after being exposed to several generations of treatment. Moving on to the reproduction, we can also see that the reproduction or the net reproductive rate, it's more or less similar, but in treatment it is similar, but till F3 and F4 generation. After, we see that there is a slight variation when the F3 generation comes, like in the F2 generation the reproduction is higher, but in F3 generation the reproduction is lower, and in F4 the reproduction is higher. We can conclude from this that the multi-generational exposure to mancozeb, it improves the survivability, that is life expectancy at birth, of Pseudodaphnia cornuta at the tested concentration of exposure. I say the tested concentration of exposure because in a lot of published ecotoxicological work, you can see that they use very high concentrations, as it induces some kind of clear response on the organism. But we were trying to have an environmentally relevant concentration, which is why we can stick to only our concentration. And then a trend was not established in all the generations of exposure, which indicates that there is some kind of underlying trade-off, which if you remember that the life expectancy at birth, it increased, which suggests that there is an increase in the survivability, but there is a lot of fluctuation in the reproduction, which can be said that this we are speculating that this trade-off might be reproduction. Also, there is another fact that if you remember the F3 generation and the F4 generation, there were fluctuations, which could also suggest that maybe for the first few generations, the organism was somehow able to protect itself from the contaminant, because in certain literature, we see that for certain metal or heavy metal exposure, this zooplankton, what it does is that it produces metallothiones, which is basically how they store the metal or the compounds inside their body without letting it move inside their body. Like they store it in their body, but it is not able to reach to the organ that it might be able to affect. So we are guessing that in the F3 and the F4 generation, because of some reason, it was able to store the components, or maybe able to evade the effect of the contaminants to F3 and F4 generation. But after that, the cost was too much, which is why it could not, there was a visible result on the reproduction itself. So this is when we realized that are we missing something? Is there anything else which could be an underlying factor, but we are missing it because we are only accessing the life history traits, which is why the future direction is to check the bioaccumulation of the chemical, or maybe it's a manganese and zinc salt, so we can also check the heavy metals presence in the biomass of the organism. So I would like to end my presentation with this. I am grateful to the Ecosystem Ecology Lab, Central University of South Bihar, for providing the infrastructure. I am grateful to UGC NET JRF and also ISEP and Lucknow University for the opportunity. These are the references. My presentation was very quick, so if anyone has any questions, please let me know.